This is a continuation of the lecture on Chapter 9. We start here with Table 9.3, reminding us that this is lecture on Chapter 9-3. So we now begin to talk about adrenal steroids after the long discussion on the gonadal steroids, and we consider the target cells in the brain. There are two types of adrenal steroid or adrenal steroid receptors. We don't want to just call them glucocorticoid receptors because, as we'll see, the mineralocorticoid is involved as well. So we saw that there's estrogen receptor alpha, estrogen receptor beta. Now we see there's a type 1 and a type 2 adrenal steroid receptors, and they are present in the brain, of course, as well as in many peripheral tissues. So the type 1 receptors resemble the kidney mineralocorticoid receptors. They bind to both aldosterone and corticosterone with high affinity. So remember, affinity and specificity were two concepts about receptor dynamics that we have discussed already. And a low affinity for dexamethasone. Remember, we saw the dexamethasone did not displace the tritiated corticosterone in that prior figure, talking about the specificity where they calculated the Ki for corticosterone for this. So the corticosterone had the high affinity and dexamethasone had the low affinity. So in that article they may have indeed have been looking at type 1 receptors based upon the data that we observed. They do not bind to synthetic glucocorticoid agonists, RU26988 and RU28362. And there's another one, are you confused yet? Sorry about that. Just couldn't resist the joke. So the highest density is in the hippocampus, the septum, the amygdala, the dentate gyrus. These are very important cognitive areas, behavioral areas for regulation of emotion and aggression and motivation and reward and happiness and all that kind of stuff. And the lower density is found in the other brain areas. There's a low density in the anterior pituitary. Again, the focus on the negative feedback primarily. So the question asked, is the negative feedback of the adrenal steroids, in particular glucocorticoids, on the anterior pituitary corticotropes mediated through the type 1 or the type 2 receptors? They bind to aldosterone to regulate salt appetite in the brain. They're occupied when corticosteroid levels are low or at baseline. So we ask ourselves, how does this happen? When you have a high affinity, then a low concentration will be effectively bound and active at the receptor. They mediate permissive circadian action of the corticosteroids. The type 2 receptors resemble the liver glucocorticoid receptors. They have a high affinity for dexamethasone, binds to corticosterone with a lower affinity than the type 1 receptors. And they bind to synthetic glucocorticoids, RU26988 and RU28362, the opposite of what we had seen for the type 1. So pharmacokinetic distinctions, pharmacodynamic distinctions that are very important. They're widely distributed in neurons and glial cells of the brain, with the highest density in the hypothalamus, high density in the anterior pituitary, especially corticotrope cells. So immediately we're beginning to think of negative feedback effects mediated through the type 2 receptors. Obviously, because it's a low affinity, they're going to be occupied only when the corticosteroid levels are high after stress or with exogenous administration. Therefore, statuses or conditions that increase the plasma glucocorticosteroids, increase the ligation of the type 2 receptors in the hypothalamus and the anterior pituitary corticotrope cells, contributing to the negative feedback effect as we would understand it. Doesn't bind aldosterone, so there is a specificity, selectivity based upon affinity. A very low affinity of type 2 for aldosterone, a very high affinity and specificity for aldosterone in the type 1 receptors. affinities differ for corticosterone, but the specificities are relatively similar. However, the specificity of the type 2 is higher, at least relevant to the aldosterone and the synthetic glucocorticoid agonists, as well as the dexamethasone. So again, the dexamethasone didn't have an effect on that study that we looked at. So we're assuming that they looked at we're looking at predominantly the type 1 receptors. And if you go back and look at that, they're discussing in the figure legend whether they got a single model or a double model from their kinetic analysis. If it were a double model, then you'd suspect 2 receptor binding sites. And it looked like from theirs, the displacement study as well, that it was single, so they probably were only looking at the type 1 receptors. So obviously the type 2 is involved in terminating the stress activation of the neuroendocrine pathways and contributing to any behavioral changes associated with the high-level glucocorticoid response to the situation. So the adrenal steroid hormone target cells in the brain contain either type 1 or type 2. So the type 1, in summary, binds both the aldosterone and the corticosterone, higher affinity for the aldosterone, therefore higher specificity. Most plasma corticosterone is bound to transcortin. We saw that on the earlier table talking about affinity, and therefore very little might be available to bind to the type 1 receptor, but any that would become free from the transcortin could bind to it. So primarily under baseline conditions, aldosterone is occupying the type 1 receptor. The type 2 receptor, the classical glucocorticoid receptors, they bind primarily the glucocorticoids and have the highest affinity and therefore specificity for the glucocorticoids, cortisol in humans, and corticosterone or corticosterone in the rats. So why have 2 different glucocorticoid receptors in the brain? So the low concentration of the glucocorticoids regulate the circadian rhythmicity. The high concentrations regulate the stress response, as we had seen. The plasma glucocorticoid levels range from 0.5 to 100 nanomolar. So we see that there's a wide difference, big point spread, if you want to think of it that way, between the lowest levels. These are probably a little bit below baseline, but baseline would be close to this, and the very highest plasma levels. So there's a wide range of interstitial fluid concentrations, intracellular concentration of glucocorticoids, reflecting plasma glucocorticoid levels, to bind to the differentially to the 2 steroid receptors. Now let's consider some non-genomic actions of steroid hormones on neurons. We start first with this research article from September 1996 in Brain Research, titled Effects of Intrahippocampal Dexamethasone on the Level of Amino Acid Transmitters and Neuronal Excitability. From the previous discussion, we know that this would be the type 2 receptor that binds, has high affinity for the dexamethasone. So we are asking now, is there any possible non-genomic action of the steroids, in particular on neurons? And it will come about through, obviously, changes in the reactivity of the excitable membranes of the neurons, ultimately impacting firing rate and the neuronal integration. And these effects, as we will see, take place in relatively short temporal time frames, too short in many cases to be explained by changes in gene expression, in other words, genomic actions. So here in this figure, we're seeing the effect of intrahippocampal dexamethasone on extracellular levels of glutamate, glutamine, glutamate-glutamine ratios, and taurine. The perfusion media, artificial cerebrospinal fluid, contained dexamethasone after the 0-minute baseline measurement. The extracellular amino acid concentration is expressed as percentage of the baseline. The data are expressed mean plus or minus standard error of the mean. Statistical analyses were assessed by using repeated measures, ANOVA with Tukey's post hoc test, and the single asterisk means the probability is less than 0.05. So here we can take a look at these graphs. There are time on the x-axis and percent of control on the y-axis. This really shouldn't be bar graphs, they should be line graphs because it is a continuous recording from individual animals, therefore the repeated measures. I really don't like the way that the authors represented this data. It should be a line because we're following the effect of infusion of dexamethasone over time, and therefore taking the recordings from the same animals, so we're comparing these different levels within animals across this time period, and therefore you have the repeated measures. ANOVA, the Tukey's post hoc test compares each of these to each other, each column, each graph, bar on this graph with each of the others. So what we see for the glutamate is that the 30-minute time point is the most significant increase. We see that there's a decrease in glutamine and an increase in taurine. But basically from these and from the ratios, they feel that at the 30-minute time point they are detecting non-general metabolism amino acid levels of glutamate. So this they are considering, therefore, through this kind of sophisticated approach, represents an increase in synaptic concentration of glutamate released at synapses in the hippocampus. And then, of course, they proceed to perform electrophysiological measurements in the same nearby areas where they perform the microdialysis, dropping in the cannula, dialyzing into the brain the dexamethasone and dialyzing out of the brain interstitial fluid to measure the amino acid concentrations. This is figure 3. It's a representative electrophysiological recording of average population spikes. So in recording in the hippocampus with the appropriate stimulus intensity, you see the stimulus artifact here at the beginning in each of these traces, and then you see the population spike. This is produced by the cell bodies, a population of cell bodies in the recording area, depolarizing and producing this spike. It's considered to be representative of the generation of action potentials or anaction potential in a population of neurons. Okay, so if you have glucocorticoid present, dexamethasone in this case, at higher levels, then more glutamate is released, then you should be able to produce a greater level of excitation in the cells. And what they're looking at is a paired pulse type of a thing, where typically when you stimulate once and then you follow up with another stimulus, you don't get as high of an amplitude. I think if you look at the way they measure the amplitude here, basically you come across here, you don't get as high of a response in the second one as in the first at 0 minutes, but at 60 minutes they are nearly the same. So this basically is what they're measuring. Can you get a second response of the same intensity because additional glutamate is being released due to the dexamethasone being present? So in putting this on a timeline, then we see in this graph the time in minutes and the percent of control and response of the population spike and of the glutamate concentrations, and there's a tight correlation at the 30-minute time point of the elevated glutamate released due to the dexamethasone, and actually there is no effect on the population spike. There's a population spike inhibition later, even though the glutamate levels are normal. So what they're predicting then is if you administer glucocorticoids, you blunt the excitation in the hippocampus. So at first we thought that this 30-minute increase might be significant, but indeed it is not. There's actually an inhibition of the excitation. And we can see that in reinforced in this panel here, figure 912 from our textbook, the effects of corticosterone injected intraperitoneally in ethanol on the electrophysiological activity of single nerve cells in the hippocampus of a freely moving hypophysectomized female rat. Now, why they had to be hypophysectomized, it's pretty dramatic removal of all the pituitary hormonal systems, but certainly would get rid of endogenous corticosterone. Samples of 1 second duration from the record show the unit activity before left and after the corticosterone infusion. So we have time in hours here. This is biologic. This is an awake behaving animal with an implanted electrode, and the mean number of spikes per second, or the firing rate of a population of neurons. A trace of action potentials is shown here. So when the hypophysectomized animal is behaving, we see that there's a baseline firing rate of about 15 hertz, spikes per second. Use the ethanol vehicle control, and there's no significant effect on the firing rate. Add the corticosterone, 1 milligram, and as plasma levels increase over nearly an hour, the firing rate is suppressed significantly down to just maybe an average of about 3 or 4 hertz. So this evidence does correlate with this here, even though at 30 minutes there's an increase in glutamate release. There is a suppression of the excitability of the neurons that extends out, in this case, up to 3 hours. And here we're seeing it also about 1 to 3 hours. And this is a trace of the action potentials. We also see cortisol affecting other neuron populations in the peripheral nervous system. And in this case, first we look at the trace. This is membrane potential. You see that it's measuring the resting membrane potential at about minus 60 millivolts. Remember, we said it's not always minus 70 for neurons everywhere. In this case, it is a guinea pig celiac ganglion, part of the sympathetic nervous system. So the resting membrane potential is about minus 60 millivolts. That's fine. About 10 or 15 or 20 millivolts positive from that, near or above the minus 50 would be threshold to fire an action potential. You can see that they're administering hyperpolarizing stimuli on a regular basis here. This is a time scale of 60 seconds. So 2 or 3 of these is a minute, so it might be 1 every 20 seconds. I'm not really sure why they needed to do that, but that's what these downward deflections are, is hyperpolarizing events. When the cortisol is added, you can see that there is a hyperpolarization of the resting membrane potential in general, making these neurons less excitable, similar to the blunting of excitability in the hippocampus. So this would be consistent with turning off neuronal drive to the hypothalamic-pituitary adrenal axis for a modulation of the negative feedback during stress. In addition, it should be representing the resolution of the behavioral activation to the stressful stimulus. So this is the grand extension of that information as we see it. So why would we have glucocorticoid receptors in the nervous system obviously to contribute to the coordination of an appropriate neuroendocrine and behavioral and psychogenic response to a stressful stimulus. We want to heighten everything, and then as the hormones 3 reach a high level, we want to start bringing everything down to normal or contribute to the de-escalation of the excited stage of the organism. This now is figure 913, showing a different way of looking at the effects, in this case, of gonadal hormones on neuronal activity. Excuse me. And in this case, on the x-axis, we're looking at the number of cells recorded per 10 penetrations. So this is the comparison of the number of active cells in different neural areas in estradiol benzoate treated and untreated ovarectomized female rats. So again, ovarectomized to eliminate the estrogen, and you can treat some of them with that, and then you get the two states: estrogen absent, basically, estrogen present, and it also, of course, stabilizes the estrogen levels in the female rats, so you don't have to worry about the stage dependency throughout the estrous cycle. Female rat behavior changes very dramatically, reproductive behavior and maternal behaviors, and therefore the female rat is studied most commonly. And these are certain areas of the brain: the bed nucleus of the stria terminalis, just in front of the lateral hypothalamus, the medial preoptic area, the medial anterior hypothalamic area, and the basomedial hypothalamic area. So we recognize at least these three from our studies of the hypothalamus. This is part of the limbic system just near the septum in front of the hypothalamus, and it's a very important reproductive behavioral area, high in concentration, high in density of estrogen concentrating neurons. Estrogen receptor expression is very high in this area. So therefore, you get a significant increase. So we've got 2 asterisks here. That's p less than 0.01. They're not showing the error bars, but we see that there is a robust difference. Our scale is from 0 to 50, so it's 10, 20, 30, 40, 50. So we're going from about 10 to about 25, so an increase of about 15 neurons per 10 penetrations into the brain. And this is in the intact animal, the brain in situ. And we see that the estradiol also depresses neuronal activity, decreases the likelihood of finding a spontaneously active neuron. I don't know what the firing rate cutoff was on this, but it decreases it significantly in the bed nucleus of the stria terminalis, the medial preoptic area, and no significant effect, as we would suspect here, in the medial anterior hypothalamic area, and a significant increase in the basomedial hypothalamic area. So there is a significant short-term effect, non-genomic effect, that might be occurring here. Now what we would have to ask ourselves, this was published in 1976, what we'd have to ask ourselves is how long were the animals administered the estradiol benzoate? Was it an acute injection with immediate recording, or were they injected over a period of days so that might express a genomic effect, change in gene expression of receptors? We saw the effect of hormones on the muscarinic receptor before. That's a cholinergic receptor. So it's possible, depending upon the experimental design, that this could be a genomic effect on receptors or ion channels if the overactomized animals were treated with the estradiol benzoate for a period of hours, 12, 15 hours or days before they made the measurements. If it was more immediate, such as here, within just an hour or so of the immediate injection, or even here, then we would think that would certainly be non-genomic effect. So we would need to know the time scale for the estradiol benzoate treatment to really appreciate this as a non-genomic effect, but definitely effects on neuron excitability. And again, we see it here in figure 914. Another way of looking at neuronal activity, spikes per minute. So this is not in hertz. This is spikes over 60-second time period or minute. So we have minutes and after progesterone injection on the x-axis in both panel A for the anterior hypothalamus and panel B for the preoptic area. So what we can see, let's check first. So it's changes in single cell activity, looking at just one single cells following progesterone injection at time 0, the increase in firing rate of a single cell in the anterior hypothalamus and the decrease in the firing rate of a single cell in the preoptic area. These figures are showing just one cell. We would hope in the full study they measured more than just one cell in several animals. The decrease in the firing rate of a single cell in the preoptic area after the intravenous infusion of 400 micrograms of progesterone in saline. The control injections had no effect on the firing rate in these cells. So we don't know if this is male or female, and if there's any type of alteration of the natural state of the HPG axis. But what we can see is that in the anterior hypothalamus, when you have a relatively low firing rate in neuron, the progesterone, if it belongs as a progesterone responsive neuron, increases its firing rate dramatically to, if it hits 60, and that's 60 spikes in a minute, that's just 1 hertz. So this isn't a gigantic increase in activity. These are, if my mathematics is right, these are pretty low levels of neuronal activity. Still, there is the possibility for a dramatic increase in firing rate, and the saline washes out, and the saline has no effect, the vehicle control. And in the preoptic area, you get the opposite effect of high firing rate. So if it was 240 divided by 60, that would be 4 hertz. So 3 to 4 hertz, I guess we would say, activity, and decreases almost to 0 as an effect of the progesterone, and within 20 or 30 minutes. So this is too fast to have any genomic effect in both of the cases. So we ask the question, where is the receptor responsible for this short-term effect? And of course, now they're beginning to detect proteins in the plasma membranes that can bind these steroid hormones. We still don't know if they're functional receptors and are responsible for this. A lot more studies need to be performed. But basically, we still accept the fact that this is one way of viewing immediate effects of steroid hormones on neuronal activity for neuroendocrine function, for behavioral function, for metabolic function as far as affecting the autonomic nervous system and other metabolic pathways. You know, the hypothalamus involved in food intake and basal metabolic rate and all those types of things. So it can be a short-term effect, and then through the genomic mechanisms, a more long-term and persistent effect. This slide is here to remind me that this chapter includes discussion of the thyroid hormone receptor mechanisms. We will reserve discussion of that until we discuss in detail the HPT